Hello and welcome to Chapter 4, Sensation and Perception. Sensation and perception are very hard to separate because people automatically start organizing incoming sensory stimulation the moment it arrives. For theory and research, however, the distinction between the two is very useful. So sensation is the stimulation of sense organs, for example, absorbing energy of the light through your eyes. And perception is the selection, organization, and interpretation of sensory input, translating the sensory input into something meaningful. Look at the photo of Rose in your textbook, and your eyes sense the light reflecting from the page, and what you perceive, however, is the picture of the rose. Psychophysics is the study of how physical stimuli are translated into psychological experience. Thus, psychologists in this area are interested mainly in sensation and perception. Synthesia is the condition where perceptual or cognitive activities such as listening to music or reading cause exceptional experiences where the experience of senses become paired. For example, words and color or sounds and visions or emotional experiences. Sensation begins with a detectable stimulus, but what counts as detectable depends on what or who is doing the detecting. Gustav Fechner in 1860 questioned, for any given sense, what is the weakest detectable stimulus? The concept of the threshold is implicit in Fechner's question. A threshold is defined in the text as a dividing point between energy levels that do or do not have a detectable effect. So, for example, automatic lights turn on when a threshold is reached. An absolute threshold is the minimal amount of stimulation that an organism can detect. It depends on the boundaries of an organism's sensory capabilities, and researchers discovered, however, that there's no single stimulus intensity that results in a jump from no detection to 100% detection every time. Thus, researchers use the concept of absolute threshold, the stimulus intensity that can be detected 50% of the time. Fechner was also interested in the smallest difference in the amount of stimulation that a specific sense can detect, the just noticeable difference, the JND, smallest difference detectable. Research on the JND by Ernst Weber illustrated that the size of the JND is a constant proportion of the size of the initial stimulus. In general, stimulus increase in magnitude and the JND becomes larger. So in terms of examples of absolute threshold and the sense involved, vision, a candlelight can be seen at 50 kilometers on a clear, dark night. For hearing, a tick of a watch under quiet conditions at 6 meters. For taste, it's 5 millimeters of sugar and 7.5 liters of water. For smell, it's 1 drop of perfume diffused into the entire volume of a 3-room apartment. And for touch, it's the wing of a fly on your cheek from a distance of 1 centimeter. So, being asked whether one light that has twice the energy of another is twice as bright is an example of scaling. And Fechner's law asserts that larger and larger increases in stimulus intensity are required to produce JNDs in the magnitude of sensation. Signal detection theory holds that detection of sensory information is influenced by two things. One, noise in the system, so irrelevant stimuli in the environment that elicit neural activity. And two, decision-making processes. So that signal detection theory was important in that it emphasized factors other than stimulus intensity, the influencing detectability. This is in contrast to Fechner's ideas. So many researchers using very different methods have demonstrated that perception can occur without awareness, and that Many people believe that advertisers attempt to influence and place subliminal messaging or messages in ads, while others say that people are just reading the ads themselves or things into the ads, like seeing familiar shapes in the clouds. Regardless, research really just shows that the effects of subliminal perception are relatively weak, and that it has little practical impact. Prolonged stimulation may lead to sensory adaptation or a decline in sensitivity to the actual stimulus. So, for example, you don't smell the skunk that sprayed you yesterday, but everyone else does, or the pool is cold for the first time you entered it, and then you become more comfortable with the cold water. So if we look at figure 4.2, there are some possible outcomes in signal detection theory. This particular diagram shows 4 outcomes that are possible in attempting to detect the presence of a very weak signal. The criterion you set for how confident you want to feel before reporting a signal will affect your responding. So for example, if you require high confidence before reporting a signal, you will minimize false alarms, but if you'll be more likely to miss some signals. So let's look at light. Light is electromagnetic radiation that travels a wave. The wave travels quickly, and we talk about the speed of light. Light waves vary in height and amplitude, which affects brightness, and in wavelength, which affects color, and of distance between peaks. Light can also vary in its purity, which has to do with how many different wavelengths are mixed together, and purity influences the perception of saturation or richness in colors. The eye has 2 main purposes. It provides a house for the neural tissue that receives light, the retina, and channeling light towards the retina. The eye is composed of the cornea, a transparent window where light enters the eye, and the lens, which is a crystalline structure that lies right behind the cornea and focuses the light rays on the retina. The iris is the colored ring of muscle around the pupil, the black center of the eye, which constricts or dilates depending on the amount of light present in the environment and changes the size of the pupil. The size of the pupil regulates the amount of light by constricting to let in less light, and vice versa. In nearsightedness, you see things clearly close up, but they are blurry far away, and farsightedness is the opposite. Saccades are constant, tiny, nearly imperceivable eye movements that help to make vision clear by briefly fixating on certain points as the eye scans the environment. The retina is a piece of neural tissue that lies behind the back of the eye. It absorbs light, processes images, and sends information to the brain. The fova, a tiny spot in the center of optic disc, contains only cones. This is where visual acuity is greatest. Axons from the retina to the brain converge at the optic disc, a hole in the retina where the optic nerve leaves the eye. If an image falls on this hole, it can't be seen. This is called the blind spot. The visual receptor cells in the axon are the rods for black and white and low light vision, and the cones for color and daylight vision. Adaptation and becoming more or less sensitive to light as needed occurs in part due to chemical changes in the rods and cones. Receptive fields are the collection of rods and cones, receptors that funnel signals to a particular visual cell in the retina. Lateral antagonism or lateral inhibition occurs when neural activity in a cell opposes activity in surrounding cells. Visual information processing, light striking the rods and cones, triggers neural signals to move to bipolar cells, then to ganglion cells, and then along the optic nerve to the optic chiasm, where the optic nerves from the inside half of each eye cross over and project to the opposite half brain. This crossing ensures that signals from both eyes go to the hemispheres of the brain. After the crossing, 2 visual pathways exist. The main pathway goes through the lateral geniculate nucleus in the thalamus and onto the primary visual cortex in the occipital lobe. The other goes through the superior calculus to the thalamus and on to the primary visual cortex. The main visual pathway is divided into two subspecialty pathways, the magnocellular channel and the parvocellular channel. These channels engage in parallel processing, which involves simultaneously extracting different kinds of information from the same point, with the parvocellular channel handling perception of color, for example, and the magnocellular channel handling brightness. In the early 1960s, researchers Hubble and Wiesel started research using microelectrodes recording of axons on the primary visual cortex of animals. Initially, they had little success getting neurons to fire by having cats look at flashing spots of light. But accidentally, they introduced a straight line and rapid firing occurred in the visual cortex. They went on to discover that visual cortex has feature detectors in it, neurons that respond selectively to very specific features of complex stimuli, such as lines and edges. This was groundbreaking research which won them a Nobel Prize in 1981. Later research in the area demonstrated that there are cells in the temporal lobes of monkeys and humans along the visual pathway that are specifically respond to pictures of faces, grandmother cells. Visual agnosia is an inability to recognize objects that are clearly visible, or prospagnosia is the ability to recognize faces. Color is largely a function of wavelength. Lights with the longest wavelengths appear in red, and while those with the shortest appear violet. Amplitude is related to brightness, and purity to saturation of color. In the mid-1800s, Young and Humboldt came up with the first theory of color vision, a trichromatic theory. This theory holds that the human eye has three types of perceptors with different sensitivities to different light wavelengths. One for red, one for green, and one for blue. And all colors can be seen according to this theory because of color mixing. But what about yellow? Is it just reddish-green? Well, Edward Herring in 1878 proposed opponent process theory, which holds that color perception depends on receptors that make antagonistic responses to three parts of colors: red on green off, yellow on blue off, and black on white off. This is not only takes care of yellow, but also explains the phenomenon of complementary afterimages. While researchers argue about which one is right for almost about a century, most psychologists now agree that it takes both theories to explain color vision.